Hi friends, welcome back to the channel. In today's video, we will dive into molecular biology math focusing on percentage solution, one of the most common calculations used daily in the lab for preparing buffers, dilutions and reagents. So let's get started. Why percentage dilution matters? Percentage dilution matter because they define the exact chemical environment required for biological reactions to work correctly. Even small errors in percentage in concentration can inhibit enzymes, disrupt cell integrity, alter reaction efficiency, and make experiments irreproducible. Accurate percentage dilutions ensure consistent buffer composition, reliable assay performance, and high quality reproducible data across molecular biology workflows. Percentage simply means parts per hundred. In the context of solutions, a percentage value tells you how much solute is present in every 100 ml or 100 grams of the final mixture. For example, a 10% solution contains 10 parts of the solute in a total of 100 parts of solution. Understanding percentage as a ratio of 100 makes it easy to prepare, dilute and compare solution concentration accurately in molecular biology experiments. When you see something like 70% ethanol or 10% glucose, the percentage always means parts per 100. So 10% means 10 out of every 100. 70% ethanol means 70% 70 ml ethanol in every 100 ml of final solution. 5% NaCl weight per volume means 5 gram of NaCl in every 100 ml of the final solution. This definition is the foundation of everything we do next. When preparing a diluted percentage solution from more concentrated stock, we use the proportional equation. Stock percentage by 100 into X by final volume equal to desired percentage divided by 100. This works because both sides describe a fraction contributed by the stock equal to the fraction needed in the final mix, whereas X is the volume of stock solution needed and final volume is the total desired volume. Now let's take some example problems. First problem is prepare 20 ml of the 70% ethanol from a 95% stock. So here the stock concentration of ethanol is 95% and we want to make 70% ethanol of 20 ml. So here is the equation, stock percentage by 100 into X by final volume equal to desired percentage divided by 100. So substituting all the values from the question, so 95% is the stock, so 95 by 100 into XML. So XML is the final required volume of 95% stock to prepare the 20 ml volume equal to 70% of the desired concentration. So solving for X, we will get X is equal to 14.7. So that is the amount of 95% stock that is required to prepare 20 ml of 70% ethanol, and combining 14.7 ml of 95% ethanol stock with 5.3 ml of distilled water, you will get 20 ml of 70% ethanol. So just go through this calculation and you will understand how to calculate 20 ml of 70% ethanol from the stock. You can vary this concentration and practice yourself to get more understanding of this problem. So this is a common problem that is used uh, daily in lab for preparing different stock solutions. So you can play around with the numbers to get the correct answer. To get the different calculation for practicing. Now let's look at another problem. So if 20 gram of NaCl are dissolved into final volume of 500 ml, what is the percentage concentration of NaCl in the solution? So the concentration of NaCl is 20 gram per 500 ml weight per volume. To determine the percentage weight by volume of the solution, we need to know how many grams of NaCl are in 100 ml. So we can set up an equation of two ratios in which X represents the unknown number of the grams. This relationship is read as X gram is to 100 ml as 20 gram is to 500 ml. So solving for X gives us the following results. So X gram equal to 20 gram into 100 ml divided by 500 ml. Solving for this, we will get 4 grams. So therefore, there are 4 gram of NaCl in 100 ml. That is 4 gram per 100 ml, which is equivalent to a 4% solution. So The answer is 4% of the solution. So you can vary these numbers and practice yourself to get more understanding. Let's look at another problem. If 6 ml of distilled water is added to 4 ml of 95% ethanol, what is the concentration of diluted ethanol solution? The total volume of solution is 6 ml plus 4, that is 10 ml. This volume should appear as denominator on the left side of the equation. This dilution is same as if 4 ml of 95% ethanol were added to 6 ml of water. Either way, it is a quantity of 95% ethanol stock that is used to make the dilution. The 4 ml therefore should appear as the numerator in the volume expression on the left side of the equation. So substituting that into the formula, 95 by 100 into 4 ml by 10 ml equal to x by 100. Solving for x, we get 38. So if x in the original equation is replaced by 38, it is seen that the new concentration of ethanol in the diluted sample is 38%. So if 6 ml of distilled water is added to 4 ml of the 95% ethanol, the final concentration of ethanol in 10 ml would be 38%. So it has reduced from 95% to 38%. So let's look at another problem. How many microliters of 20% SDS are required to bring 1.5 ml of solution to 0.5%? So it's a little bit tricky question. So here we substitute all the values. 20% SDS, that is 20 divided by 100 into x ml divided by 1.5 ml plus x ml, that is equal to 0.5% solution, that is 0.5 by 100. So solving for x, we will get 0.03846 ml. So this is given in ml. Now we have to convert into microliter because actually the question asks for microliter. So to convert this into microliters, just simply multiply 1000, then you will get 38.5 microliter. So therefore, if 38.5 microliter of 20% SDS are added to 1.5 ml, the SDS concentration of the sample will be 0.5% of the final volume of 1.5385 ml. And if there were some other components in the initial 1.5 ml, the concentration of that component would change by the addition of SDS. So this is how we solve addition of component to already existing solution to make a particular percentage solution. So what are the key takeaways? So the key takeaways are percentage always means per 100 ml or per 100 grams. Always include final volume in your calculation. When you add stock, the final volume changes, and don't forget this. Use simple proportional ratios to solve the dilution problems, and these calculations underpin almost every protocol in molecular biology. You know that every day we use different ratios of buffers, calculation requirement for buffers and enzymatic reactions and other reagent preparations. So these will come handy when you practice it. It will be much more easier and you don't make mistake. Hope you enjoyed the video. Thank you for listening. If you like the video, please press the like button and share it with your friends, and subscribe to the channel for more useful contents. Thank you.